Hello, Dr. Jamil. Welcome.
Hi, pleasure to be here.
So how is it in Ashoka? Have you?
Yeah, I joined Ashoka. I'm right now. I'm at home, but
yeah, it's all online.
yeah.
Hi, Jam. How are you?
Hi. TRG, why don't you put the video on and introduce this is Professor Govindarajan from IMSC Institute of Mathematical Sciences, also Dr. Jamil. I think Vijay.
Hello, sir.
You want to switch on my video?
Yeah, just anyway. I think you've already met. Vijay, YouTube link is recording in India.
Yeah. Sundar, can you put the YouTube link here?
Yeah, in the chat box.
I think Sundar, do you have the link, or I can pick up the link from YouTube? It's very much available in the YouTube now.
Yeah, I'm posting.
Okay. Dr. Jamil, your mic is muted.
Okay, let me just check. No, it's not. Okay, you can't hear me?
Yeah, we are hearing you now.
Yeah. I'm just trying to minimize this this thing at the bottom because it's really coming in the way. Can anyone help me with this? See all the participants I'm seeing at the bottom. How do I get rid of that?
Yeah. Okay. To the top right, you will see 3 symbols.
Right.
And you can choose whichever are the size. You choose the least of the 3, then the panel should go.
Record, meeting info.
No. To the top right, there will be 3 symbols.
3 dots.
3 rectangles, yeah?
Oh, 3 rectangles. Let me escape and then do that. There's full screen, there's gallery, and there's speaker.
No. When you share screen, you will get 3 horizontal rectangles on top.
After I share the screen?
Yeah. Stop right. And those let you adjust the size of the panel. You see?
I don't see.
Oh, I see. What machine is this?
Oh, it's a Mac.
Can you just give me one moment? I will.
I got it. I got it. Done. It's done.
Yeah. Okay. Is it okay now?
Yeah. All right. Wonderful. Sorry about the delay. I will be speaking about the 2020 Nobel in Physiology or Medicine, which was given to 3 scientists who worked in US for the discovery of hepatitis C virus. Harvey Alter, who worked for over 50 years at the NIH in US. Michael Houghton, who was working at the Chiron Corporation, which was a biotech company in the Bay Area, San Francisco Bay Area, when he made the discovery, but later he has moved to Canada at the University of Edmonton. And the third person is Charles Rice, who at the time of his initial discovery was at Washington University in St. Louis. And now he's at the Rockefeller University in New York. So what is viral hepatitis? Hepatitis stands for inflammation of the liver. The inflammation of the liver can be due to multiple reasons. And one of those reasons, one of the major reasons, is a viral infection. Other reasons are alcoholic hepatitis. For people who drink too much alcohol can get hepatitis. When fat accumulates in the liver, that's called fatty liver, and that also causes hepatitis. But viral hepatitis is a major cause. And in the 1950s, people realized that there were 2 kinds of viral hepatitis. One was called infectious hepatitis, and the other was called serum hepatitis. Serum hepatitis because this could be transmitted from contaminated blood. And it was later found to not be associated with blood cells, but rather with the fluid form of blood. So that's why serum hepatitis. The second type was infectious hepatitis, meaning that it could be transmitted directly from a person to the other, one person to the other through casual contact. And later in the 1970s, the very first hepatitis virus to be discovered was called the hepatitis A virus, and that's how the alphabet started. It was discovered by a man named Stanley Feinstein through electron microscopy, and Robert Purcell, both at NIH. Subsequent to that, in the 1950s, work had started on serum hepatitis. And later in the 70s, or rather late 60s, Baruch Bloomberg discovered the hepatitis B virus. And that's another story. And I'll come to that in a minute, because that is intimately linked to the discovery of hepatitis C virus. But after hepatitis B was discovered, people realized that even though blood was being screened for hepatitis B virus, they were still getting viral hepatitis associated with blood transfusions. And that's when in the 1970s, this term came about, which was called non-A, non-B hepatitis, meaning that these people had hepatitis, but it was neither the A type nor the B type. And later in the 1980s, the hepatitis C virus was discovered, and that's the topic for today, and I'll go through how it was discovered. And in the early 90s, although we had known about this agent since the 70s, the hepatitis E virus was discovered. And my lab essentially spent 25 years working on the hepatitis E virus while I was at ICGB. Okay, so let's start this with why hepatitis viruses are important. Although there are 5 hepatitis viruses known at this time, called hepatitis A, B, C, D, and E, they are not belonging to the same family, or they don't have the same, they're not the same type of viruses. So for example, hepatitis A, C, and E are RNA viruses, all belonging to different families of RNA viruses. Hepatitis B is a DNA virus, which is very similar to retroviruses because it also involves retrotranscription in its life cycle, or retrotranscription means converting RNA into DNA. And hepatitis D virus is not actually a virus, it's a viroid, meaning that it can't replicate by itself and it needs another virus infection to help it replicate. But if you look at this, at the load of viral hepatitis around the world, and this slide is a bit older, when the world population used to be about 6 billion, now it's close to 8 billion. You could see that a lot of people around the world historically have been infected with these viruses. Hepatitis B and C also become chronic infections, whereas A and E do not become chronic infections. So that's the reason why these viruses are important and so many people work on them. Now, hepatitis B, I mean, this is just a reiteration. Hepatitis B, about 2 billion people have been infected the world over. More than 240 million worldwide are chronically infected, meaning that they have not been rid of the virus. They carry the virus in them, although the virus may not be showing disease. But these people are capable of transmitting the virus to somebody else. And annually, somewhere between 500,000 to 700,000 people die of hepatitis B infection. To put this into perspective, that's about half the people every year that COVID has killed so far So keep that in mind. Hepatitis C, on the other hand, some 1.5 billion people, almost same as hepatitis B, are chronically infected. So this virus has a potency to become far more chronic than hepatitis B does. And again, more than 350,000 people are estimated to die of this virus every year. Now, I'll talk about hepatitis B first because that makes a nice story when we come to the Hep C virus. This was really the first blood-borne virus to have been discovered. And it was discovered by a man named Baruch Blumberg, who was at that time at the Fox Chase Cancer Research Center in Philadelphia. And for this discovery, he got the Nobel Prize in Physiology and Medicine in 1976. Those of you who are interested in reading books, Baruch Blumberg has written a very nice book. This is a few years old on the hunt for this virus, and you may be able to still find it in libraries or Amazon or whatever. On the right, I show you a picture of what the virus looks like. And if you look at this picture closely, you will see that there are three types of virus particles in this picture. You have these small rounded particles, you have the elongated particles, and then you have the large round particles which seem to have a core in them. The ones with the core in them are the actual virus. The small round particles and the elongated particles are actually empty shells. They are called virus-like particles. They're just like the virus, but they are missing the genetic material. And this is produced in plenty in the blood of infected people. So Baruch Blumberg was actually looking at something that he called the Australia antigen because he discovered it in the blood of Australian Aborigines. And he was looking for something completely different, but then he correlated the appearance of this protein in the blood of people with hepatitis. And it turned out that it was the hepatitis B virus. So a very serendipitous discovery led to the first blood-borne virus. But that was not just it, because when people looked at the prevalence of hepatitis B carriers around the world, shown on the world map on the left, it matched very well with the annual incidence of liver cancers shown on the right. And this led to the hypothesis that hepatitis B infection of the liver may be one major cause of liver cancers. And this turned out to be true. Hepatitis B chronic infection leads to a long-term sequence of events that leads to cancer of the liver, sometimes 20 or 30 years after the first infection with the virus. And very quickly, a vaccine was prepared against hepatitis B virus. And that is the reason I showed you this picture of blood from hepatitis B carriers. Essentially, plasma was taken from hepatitis B infected people. These empty particles were purified, and these were first given as a vaccine. These were, remember, these were just virus-like particles with no DNA, so they were not infectious. And this was the first vaccine that came, which was called a plasma-derived vaccine. And for many years, people were vaccinated with the plasma-derived vaccine. However, with the advent of recombinant DNA technology in the early 1980s, the very first recombinant DNA vaccine which was made was against hepatitis B virus. Scientists took the gene for the surface protein of the virus, expressed this protein in yeast cells, and in yeast cells, when you express this protein, it forms virus-like particles just like the empty particles you see in blood. So it was fairly easy. You took the particles from yeast, you purified them, and you formulated those as a vaccine. So this was really the first instance of not just a vaccine against a blood-borne human virus, but it was also actually the first human cancer vaccine. The next one, obviously, is the human papillomavirus, which causes uterine cancer, for which, again, a Nobel Prize was given many years ago. All right, so this brings us nicely to what Harvey Alter discovered. So in the 1950s, blood transfusion that people got during operations was a very risky business because 1 in 3 people who got blood transfusions got some kind of hepatitis. And Harvey Alter, who was working at the NIH Blood Bank, actually worked with Baruch Blumberg and supplied him the blood from which he made some of the hepatitis B discovery. So Harvey Alter, of course, was not awarded at that time. It was only Blumberg who got the prize for hepatitis B discovery. But this work led to the development of serological tests against hepatitis B. And people realized that by the mid-70s, when tests were available against hepatitis B, 80% of the post-transfusion hepatitis was a non-A, non-B type of hepatitis. Harvey Alter's contribution was that he very systematically took serum from people who had non-A, non-B hepatitis, injected this serum into chimpanzees, and showed that you could produce human-like disease in chimpanzees. And then later he showed that the agent present in serum was actually a virus. So that was his original contribution, coming from the discovery of hepatitis B to the development of tests against hepatitis B, recognizing that there was a lot of post-transfusion hepatitis that was non-B type, and then using the non-B serum to show disease in an animal model and show that it was due to a virus. All right. Now, coming to Michael Houghton, Michael Houghton's contribution was that he actually found the virus. So while Harvey Alter, with his experiments, showed that the agent was a virus, he didn't know what the virus was. Michael Houghton started working on this in 1982 to actually find the virus. And first he tried to find the virus protein in blood, just like Baruch Blumberg had tried to find antigen in blood. However, in this case, he failed because unlike hepatitis B, which produces a lot of protein in blood, hepatitis C does not. It's a different virus. It does not put out a large amount of protein in blood. So the next thing that Houghton tried was to use an approach, a recombinant DNA approach of making cDNA libraries or copy DNA expression libraries from patient serum. So he purified RNA from the sera of patients who had non-B hepatitis. He converted the entire RNA into DNA, put this DNA into vectors which could express protein out of this, and asked if bacteria would express these proteins. And then he would detect those proteins using serum from patients, the idea being that patient serum contained antibodies to whatever was infecting them. And this was really, really tiresome work. Remember, he started in 1982, and only about 6 or 7 years later, he was able to show after the screening about 2 million clones that he found one positive, which reacted to the to the serum from patients. And this piece of DNA actually came from this new virus. And this he published 2 back-to-back papers in Science in 1989. One was the isolation of a cDNA clone, and the second paper was to develop an assay which looked for circulating antibodies to non-A, non-B hepatitis. So now there was a tool available and there was a test available to test the blood supply against this non-B hepatitis, which was later called the hepatitis C virus. The work for which Charles Rice was awarded was to show that he could make the virus replicate. The virus had been identified. There was a name to it. It was called HCV. Scientists knew that the genome of this virus is RNA. They had cloned the genome, but nobody could make it replicate. And unless you make the virus replicate, how do you develop drugs against it? How do you make vaccines against it? That was a problem. So the first thing that Charlie Rice tried was that he saw that the genome at the 3' end especially had about 100 base pairs missing. So the clone that Michael Houghton's group had made was not really a full clone. So the first thing Charlie Rice's group did was to assemble the full viral genome. And then he followed the same strategy, took this genome and injected into chimpanzees, but he found no replication. And then he realized that whatever he is using, it may have accumulated mutations over time. So that is why the genome is not efficiently replicated. So base by base, he created a synthetic viral sequence. And this viral sequence is roughly 10,000 nucleotides. So base by base, they created, based on the consensus sequence present at a given location, created this full-length genome, put it back into chimps, and now it replicated. So he had a virus, and this virus was replicated. And the reason this was important is that this discovery is the one that actually led to in vitro screening methods that were later used to produce drugs against hepatitis C. So I'll just summarize here what happened between Harvey Alter and Charles Rice. On the left, you see chimp experiments from Harvey Alter, where he injected the serum from non-B hepatitis patients into chimpanzees. And you can see that the chimpanzees gave rise to liver enzymes, showing that the liver was getting injured. About 2 decades later, Charles Rice creates a full-length clone of the virus, injects that clone into the livers of chimpanzees, and could produce the same effect, rise in aminotransferases, liver enzymes, with a clone that Harvey Alter did in 1978 using serum. So this really completed the discovery of the Hep C virus. Immediately after Charles Rice did his work, in 1999, a group at the University of Heidelberg showed that you could replicate this now in a cell line. Remember, Charlie Rice's work was about replicating it in chimpanzees, but that was not going to be very useful because chimpanzees are not a very easy model to work with. So Barton Schlager's group in Heidelberg was the first one to show that you can actually replicate this in a cell line But what he used was not a full genome clone, he used a subgenomic clone. And immediately after that, Charles Rice and his group also showed that you can now take the full-length genome and replicate it in a cell culture model by making slight tweaks in the genome. And what they also showed that the virus that they were now making in cell cultures was responsive to interferon. It was inhibited by interferon. And the significance of this is that interferon was the drug of choice for people who had hepatitis C infection or people who had hepatitis B infection. So this really completes the cycle. Now, a little bit about the virus. The hepatitis C virus is an enveloped virus. It belongs to a family of viruses which is called Flaviviridae. Flavivirus, there are many other important flaviviruses. You have dengue virus, which is a flavivirus. You have the West Nile virus, a flavivirus. A yellow fever virus is a flavivirus. So there are many flaviviruses that are linked to human disease. And the work of all of these scientists and many more showed that this is an RNA virus, has many proteins that it can code from a genome that is roughly 10 kilobases. And at the 5' and 3' ends, there are these highly secondary structure rich sequences. It was the 3' sequence which was missing from Michael Houghton's clones, which Charlie Rice realized, added, and got everything to work. And finally, when everything came together and a cell culture model was established, this really led to the discovery of two very important drugs. One is called sofosbuvir, and the other is called ledipasvir. And these are now given as a combination drug therapy against Hep C virus. The first one is directed against this protein called NS5B. NS5B makes the replicase, the enzyme that replicates the viral genome. And this ledipasvir is against this NS5A protein, which is a protein that is required for the virus to control the host cell environment to really be able to replicate in it. So all these discoveries came together, developed, and led to the development of a test that is now routinely used in blood banks to make the blood supply safer, and development of drugs, which are really the first time. These are called directly acting antivirals. These are drugs that can finally cure the virus. See, remember, this is a chronic viral infection. The virus remains, doesn't go away from the system, like in hepatitis B. Can Mr. or Ms. Natarajan mute yourself, please? So now we have these drugs which are called directly acting antivirals, which are able to cure a person of hepatitis C. We still don't have a drug that can cure chronic carriers of hepatitis B, but we now have drugs that can cure hepatitis C. And it is for this reason that work on a vaccine for hepatitis C has really fallen back. It's a difficult vaccine to make simply because the virus mutates very fast. But now that drugs are available, vaccines are no longer going to be, sort of very, very fashionable to make or very useful to make for this virus. So to summarize, the 3 Nobel Prizes, the prize awardees this year, Harvey Alter worked in the blood bank. He used blood samples to figure out that there was a new agent other than hepatitis B, which also he had helped discover, that there was a new virus that was there, and it was called Hep C virus. Michael Houghton's work using cDNA expression libraries and really, really hard work showed that you could actually fish out the clone for the virus directly from blood. And many people don't realize that this was also the first time that a viral genome was actually discovered before a virus could ever be isolated or seen. So that was really the remarkable thing about this technology. And finally, Charlie Rice came along and produced a viral genome that could be replicated, not just in animals, but also developed a cell culture system which was used to produce drugs against this. Just to remind you that every year on 28th of July, there is something called World Hepatitis Day. And although much advance has been made in the discovery of hepatitis viruses, in the development of vaccines and tests and drugs, 9 out of 10 people living in the world today with viral hepatitis don't know that they have infection. And the simple reason is that the liver is an organ that can take a lot of insult and keep going, but suddenly one day it collapses. So this World Hepatitis Day is the day that marks the attention of the world towards this problem and urges people to get themselves tested. There are some other statistics. I'm not going to go into it, but let me just say that the hepatitis B vaccine, which is a very successful vaccine, it's also now a very inexpensive vaccine, is available readily around the world. Even then, only 42% of children in the world have access to this vaccine. More than 50% children don't have access to this vaccine. So it's one thing about discovery, it's another thing about access. Scientists make the discovery, it's the public policy, the politicians who guide us in taking this discovery forward and implementing public health policy. So with that, I will end by just saying that even though we have medicines for hepatitis C, which are a direct result of what the Nobel laureates were awarded this year, the drug is extremely expensive. It costs roughly $10,000 a year for the drug regimen in the US. However, licenses have been given to some companies in India, but it still costs about $350 to $400 for a treatment, which is still very unaffordable for most of the population of our country and other parts of the world where hepatitis C is a problem. So again, a problem of access, which hopefully will get solved sooner than later. I will stop there, and if there are any questions, I'll be happy to take questions.
Thank you very much. We have a set of questions that was posted in the chat box. Apart from that, those who wants to raise the question, they can raise the hand.
So should I just read the question and answer it, or or would somebody read them?
Whichever way it is convenient for you, we can do that.
I may not be able to track all of them, so maybe if somebody.
Jian, I think maybe you should read that.
Right. Okay. So the first question. No, we receive a email question from Aravind Badrinath even before the meeting was started. He asks, sir, how much know-how does the discovery of vaccine for one kind of virus reveal about the future mutations? How adaptive can virus vaccine be?
Well, firstly, a vaccine for one virus will not work for another virus. So, for example, a vaccine against flu will not work for COVID, or a vaccine against hepatitis B will not work against hepatitis C, because they are all very different viruses. Now, once you make a vaccine, I think the question he's asking is, what if the virus changes? So again, it depends on the virus. Hepatitis B virus doesn't really change that much. So a vaccine which has been made in the late 80s still works. Whereas a vaccine against hepatitis C or a vaccine against HIV has been very difficult to make, simply because the virus mutates fairly quickly. So if you make a vaccine against one variant, the other variant takes over fairly quickly. So it depends. I think one of the concerns maybe Mr. Arvind has is the COVID vaccine, because everyone is worried about whether the COVID vaccines that are supposed to come soon, whether they will become useless after mutation. That's unlikely to be the case, simply because coronaviruses don't mutate as quickly as some of the other RNA viruses. In fact, coronaviruses mutate the slowest of all RNA viruses. The other thing is that the major mutation that's been seen so far in the COVID virus is outside the region which is important for vaccine. But one doesn't know what will happen when a vaccine comes and a large number of people take the vaccine. Then the virus will have an evolutionary pressure to mutate. Today, the virus has no evolutionary pressure to mutate because there is no vaccine. We have to keep these considerations in mind. It varies from one virus to the other.
The second question from Kumaravel. How does the Ayurvedic medicine seriously affect the liver functions?
I can't answer that question. I don't have knowledge of Ayurveda.
The third question, sir, what is Flavivirus? Flavi from Aparna.
Flavivirus is a kind of virus. It's a family of viruses. And during the talk, I gave you example of hepatitis C being a flavivirus. Dengue is also a flavivirus. Yellow fever is a flavivirus. So there are many viruses that cause human disease. They belong to this family of viruses which are called flaviviruses. Some of these are transmitted through mosquitoes, for example, dengue, yellow fever. Others are transmitted through blood, for example, hepatitis C. So there's no common way of transmission of these viruses. They just belong to the family because their genetic organization is quite similar, and the way the genes replicate is quite similar. So that's why they are clubbed together in one family.
Next question is from Kumaravel. Is there any direct test to identify HCV?
Is there any direct test?
Yes.
Yes, of course. You can now test for viral RNA using reverse transcription PCR. You can also test the blood for antibodies to HCV, which tell whether somebody has a past history of HCV infection. And it is routinely used, the antibody test is routinely used to screen blood in blood banks. So yes, there are tests available.
So next question, Dr. Ramanujam is asking, what is consensus genome sequence?
Sorry, what is consensus genome sequence?
Yeah.
So consensus genome sequence means that, when Charles Rice was looking at this, there were many viral variants that people had discovered and they had sequenced them. So he basically created a viral genome which had uh, a consensus nucleotide at each position based on what was there in the multiple variants. So let's say you're looking at 100 variants, and at position 100, there should be a T, but in some variants it's an A. So that's how he really stitched it together to create a sequence that was a consensus of all the viral genome sequences that were known at that time.
Okay. So next question is from Ganeshan. So I have seen doctors say that fatty liver is common among Indians.
Yes.
So not worry about that.
Well, I wouldn't say don't worry about it. About 20% of Indians have fatty liver. And when fat accumulates in the liver, it leads to, which causes something called fibrosis. And fibrosis is essentially hardening of the liver. And once that happens, the liver tissue gets scarred, and that becomes a seed point for the development of liver cancers. It's a slow development process, takes a long time, sometimes 25 to 30 years. But it's not something that you shouldn't worry about. You should worry about. If you have fatty liver, then you make sure that you don't eat fatty food. You exercise enough so that the fat is not stored in your liver. Basically, it's excess fat that gets stored in your liver. And you have to get rid of this by changing your lifestyle, eating habits, exercise.
So next question is from Mr. Sankaranarayanan. He's asking, what is immunoscreen? What immunoscreen method was used by Dr. Houghton?
Right. So immunoscreen simply means that I have a bunch of, let's say I have 1 million bacterial colonies that I have to screen. And the question I'm asking is, which 1 or 2 or 10 out of these are producing the viral protein that I'm interested in? So I use serum from patients, and the thinking here is that serum from patients should have antibodies to that protein, and therefore I should be able to use these antibodies to reveal those few clones out of millions that express this protein. It's a very, very tedious process. So that's basically immunoscreen. You use antibodies and immune reagent to screen.
So next question is from Abhinesh. Nice lecture, sir, but it is quite scary to see the start you had shown. Diagnostic is one of the diagnostics of one time or frequently done. Could you please elaborate the conditions in India?
The conditions of diagnostics?
The diagnostics is of one time or frequently to done. Could you please elaborate the condition in India?
Well, I mean, in India also, the blood supply is tested for both hepatitis B and C and HIV. These are the 3. But of course, it also depends on whether you're taking the blood from Red Cross or you're, just going and taking blood from a small hospital that doesn't do screening. So that is something you should be careful with if somebody needs a transfusion. As far as individual is concerned, individual should maybe get tested once, and if you were ever infected, you will have antibodies. That at least tells you careful with your lifestyle and take steps. So, for example, to reduce fatty liver. So, for example, if you have fatty liver and you also have hepatitis C past infection, which you don't know of, then it's a problem. But if you know that you both, then you will take precautions to reduce what you can. Of course, once you get infected with hepatitis C, you can't get rid of it unless you go on those medications. But fatty liver is something you can reduce. So it helps to know. And that's why this World Hepatitis Day, and in most big cities, in Chennai, for example, they would be free in many hospitals on World Hepatitis Day. So if you're worried, get tested. At least you'll know. If you don't know, you can't control it.
Next question is from Dr. Govindarajan. Other than alcohol, is there any other food item responsible for hepatitis?
Well, oil directly, but even when you eat too much carbohydrates, I mean, that's basic biochemistry, basic physiology. If you eat too much carbohydrates, they get converted for storage into fat, and then fat is stored. So basically limit your calories. Oil, yes, but limit your calories.
Please again have one more question. Any suggestion to improve the public health issues regarding hepatitis?
Sorry, I missed that. Improve what?
Any suggestion to improve the public health issue regarding hepatitis?
Well, I mean, screening, learn about hepatitis. Much has been done. A lot of literature is available. Learn about it, and make sure, of course, that you don't get. If you need blood, don't get it from places that are not certified to be testing for these viruses. Those are the main things to think about. And of course, all these blood viruses are transmitted through shared needles. So ensure that, when you get an injection, you don't, the needles are not being reused. So those are the main things.
Next question from Dr. A. Tangaraj is asking, is there any correlation between blood group and COVID-19 infection?
I did read somewhere that some blood groups are more sort of susceptible, but I don't know what's the science behind it. And I don't think many people really know what's the science behind it. But yes, there appeared to be some correlation. Some blood groups were more prone to, not infection, but rather, worse course of disease. But I really don't know what's the science behind this. I don't think anyone knows at this point.
Another question from Kumaravel. He is asking, working in hot environment, does it influence the liver hepatitis healthiness?
Hot environment? No, it doesn't.
Yeah.
I mean, we are warm-blooded animals, which means that our body temperature is constant. So even though if it is 50 degrees outside, your body temperature keeps at 37 degrees. So it's not like if it's 50 degrees outside, your liver is at 50 degrees. It isn't.
Next question from Aparna. You mentioned that Charles Rice discovery led to the important discovery in vitro screening method. Can you brief about the method, sir?
So basically what Charles Rice did was to first show that you can take a viral RNA, a full genome viral RNA, inject it into the liver of chimpanzees and produce disease. So if you remember, I had one slide where I showed what Harvey Alter did with serum. Charles Rice, repeated exactly that experiment in chimpanzees, except that he was not injecting serum which had something, but he was injecting a highly characterized genome from the virus. So he essentially established that here was a viral genome that was capable of producing the same symptoms. And then he later developed a method by which this viral genome can replicate in cell cultures. when you replicate it in cell culture, you can then use that to screen drug molecules and see, for example, which compound will inhibit the production of viruses. So that was basically his contribution.
So that ends the questions. Is there any questions from the audience? Now we can wait for a couple of minutes, and we can, if there is no question, we can wind up the session. If anyone wants to ask the question directly, they can unmute the mic and they can speak.
Yes, please go ahead, Bhanumati.
Yeah, one more.
Yes, sir. Good evening, sir. I just want to know what is the difference between yellow fever and hepatitis C?
Well, yellow fever is caused by a very different virus. It's called the yellow fever virus. And hepatitis C is caused by the hepatitis C virus. They're completely different diseases caused by two different viruses. Yellow fever is called yellow fever because it also affects the liver and it causes bile to accumulate, and therefore the skin appears to be yellow.
Oh,
that's why it's called yellow fever.
Thank you, sir. One more thing, can I ask?
Yeah, please.
Actually, the blood transfusions and the transmission of these viruses through blood, whether it could be a hereditary factor also?
Sorry?
Sir, these things, these viruses, which may be caused or which may be transmitted by means of blood transfusion also?
Yes, it is primarily transmitted through blood.
Then, in case of contacts or in case of such transfusion?
No. You can't transmit it by touch.
No, whether it can.
This is a blood-borne virus.
Can be,
This is a blood-borne virus. It is transmitted mainly when blood mixes. So through injections, through transfusions, this is how you transmit.
Okay. Thank you, sir. Thank you.
And by the way, mosquitoes cannot transmit it.
Yeah.
So a lot of people
clearly said about the malaria and other things. Yes.
A lot of people think that blood-borne viruses can be transmitted by mosquitoes because mosquitoes suck blood from one person and then go and suck from somebody else. But mosquitoes never transmit these viruses simply because these viruses cannot replicate in the mosquito blood.
Okay.
Good evening, sir. I'm Dr. Bashira. Will it be transmitted vertically from mother to a child?
Yes, it can. Yes, absolutely.
Thank you, sir. Vertical transmission.
So in the meantime, let me ask one question from Vinida Chandrasekhar.
Yes.
She is asking, nowadays there is an increasing level of new viruses.
Well, if you see the 21st century, in the 20 years of the 21st century, we already had two pandemics, SARS in 2013 and COVID, which is going on right now. And there are many other pandemic threats. We had swine flu, we had Zika, we had Ebola. So all of these viruses that are emerging are viruses that are found in wild animal populations, and they jump from animals to humans either directly or through some intermediate animal host. So one of the main reasons for this is the increased contact between wild animals and domestic animals and wild animals and humans. And this largely happens because wild animal habitats are being cleared, forests are being cleared to produce, to develop more farmland, to develop housing projects, which brings animals and humans into close contact. So that's one of the main reasons why many of these human viruses are many of these animal viruses are jumping into humans. And of course, the other reason is food habits. When you start eating wild animals and you start catching wild animals for food, you increase the chances of viruses in animals jumping into humans. So those are basically the reasons.
Hello, sir. Can I ask a simple question?
Sure.
Yeah, I mean, it's not a question, but I just want to have a comment on this. I mean, mostly the researchers nowadays focusing on climate change impact on everything, be it water
Yes.
resources,
Yes.
water.
Yeah.
So the climate change, by climate change, what they mean, I mean, basically it's a global warming and things like that. So I want to have your comment on the impact of climate change or so on, so global warming on this, I mean, the virus or newly emerging disease, or I mean, whatever.
Right.
Yeah.
Yeah. So very good question. Normally people don't find a direct link, but actually there is a direct link. Now, let's look at viruses that are transmitted by mosquitoes. Let's say dengue virus. When the climate warms up, the mosquito that carries the dengue virus can now survive at higher latitudes. And it can survive at those latitudes for much longer during the year. Simply because these mosquitoes can survive in a range of temperatures. And if you extend this if because of global warming, the temperature in many parts of the world, which were not conducive to the mosquitoes surviving earlier, they can survive now. So that's one reason. The second thing which you really need to worry about is what is hidden below the ice caps and the glaciers. There was some interesting experiment done a few years back where people drilled into ice cores in glaciers and brought out cores which were 5,000 years old and 15,000 years old. And by genome sequencing, they showed that about 2/3 of the genomes that they recovered are not known to humans. So what are they? Are these viruses that were there when the ice age hit and they got in the core? So if our glaciers start melting, if the ice core starts melting, we may unleash some of these. And in fact, something like this happened in Alaska a few years back, not with a virus, but with a bacterium that causes the disease anthrax. Because of the ice melting, the carcass of a reindeer got exposed in Alaska, in a village near Alaska. And it turned out that this reindeer had died of anthrax. So when the carcass got exposed, many people in that village got exposed to anthrax, got sick, and a few people died. So we don't really know with global warming what else will come out. Maybe new viruses will come out that we have never seen. Our population has never seen. So that's really a very, very big worry. So those are the two things that I think climate change has a direct effect.
Thank you, sir.
So we got a couple of more questions now. Can interferon be used to reduce chances of hepatocellular carcinoma?
Yeah, so hepatocellular carcinoma essentially means liver cancer. Interferon cannot be used to reduce chances of liver cancer. Interferon has a property of inhibiting these viruses, hepatitis B and C viruses. And they were used as drugs to treat these infections, chronic infections. But interferon itself has very, so it's not really going to be a drug of choice. Now that hepatitis C drug is available, that's a much better choice. For hepatitis B, there's a very good vaccine available. And I think all of you who are listening to this, it's a very cheap vaccine. Please go and get yourself vaccinated. It's very simple. 3 shots will give you protection for 5 years. And then every 5 years, you take a booster. It's a very, very good vaccine and very cheap now.
So one more question. It's some basic science question because we are, our audience are not having accustomed with the biology. This question is genome sequencing is a lot on the news these days. Can you explain what exactly it is? You can refer some material for this.
Sure. So genome sequencing simply means that you find out the genetic code, the genetic organization of the genome base by base. It's like having a book and knowing every alphabet in the book. Unless you know every alphabet and where it is placed in the book, you will not be able to read a sentence, you will not be able to make any sense of what the book says. So that is what genome sequencing is, to find out exactly what is the sequence of the genes of the genetic material of any organism, whether it's viruses or humans. I mean, you may have heard of the Human Genome Program. Human Genome Program essentially sequenced the human genome base by base. So we know the entire 3 billion base pairs of the human genome. That's what a genome sequence is.
Thank you, sir. Another question came from participant in YouTube. She was asking, sir, my parents were affected by HIV. What is the possibility of me getting affected with HIV?
Unless you were transfused with blood from your parents, you have very little possibility of getting infected. Just living in the same house with your parents, you will not be infected. But if you're worried, get tested.
Excellent. So now all the questions are over. We can wind up the session. Before winding up the session, I will appeal all the participants to please fill up the feedback forms that's available in the Google website, and we provided the link in the chat box. Please click the link in the chat box and fill up the form and submit it. We have our senior participant activist PK Rajan is available. Let me ask PK Rajan to deliver the vote of thanks.
Hello.
Yeah.
Am I audible?
Yes.
Dear friends and Dr. Jamil, thank you all for participating in this popular science lecture. So this is going on for a long time. So this will be continue to be held on the topical issues as well as some basic sciences, which we feel to be taken to the larger public, to make the larger public much more scientific in their perception, in their understanding of the world in which they are living in. This includes the natural sciences as well as the social sciences, which are somewhat related with this. Tamil Nadu Science Forum, for the last 35 years, is striving to take science and scientific attitude to the large population of Tamil Nadu. Tamil Nadu Science Forum is having its branches almost in all the districts, and not even in the up to the district, it is having almost in all the panchayat unions. So we have many modules. One of that is popularization of science and bringing the science in its frontier level to the common public. And we have other modules which is working for popularization of science, basic sciences, elimination of unscientific beliefs, working on the intersection of science, technology, growth, development, and environment in science publications, etc. Anybody who are having some interest in science, you can find your own space and like-minded hearts in Tamil Nadu Science Forum to work with. We invite you all not just to be a spectator and an audience to the lectures which we conduct, which we will continue to conduct, but also a fellow organizers and fellow comrade in taking the science and science messages to the larger public. With these words, I thank the organizers, starting with Mr. Vijayan, and once again thank Dr. Jamil, and I thank the event month after month over a long period of time who are owed to continue this further. Thank you all for participation. Thank you very much.